Okay, today actually I'm going to focus more on the second part of this titled talk, which is chromatin, given the nature of this meeting here. Okay. John introduced a number of aspects of chromatin structure, and I'll just briefly touch on a couple of those right now. I mean, one of the age-old questions, and it as well, is do cells really care where nucleosomes are positioned? And here the age-old chromatin, which is through electron microscopy and string. These balls here are nucleosomes, and then higher order is where the nucleosomes adopt preferred positions, and you really essentially. But I want to emphasize cells really do care, because you can imagine a site on the cell being rotated, so a protein potentially can bind on it, rotate that DNA, little 5 base pairs, that site will now be facing inward to be able to bind. So, positioning is quite critical. Hopefully, with technology, we'll get at that answer. Regulatory transcriptional start sites. But now get to this structure, and we've been involved in this as well in terms of mapping. I'm going to tell you a little bit about the methodology in a second. But I just want to tell you about the cell. So the question is, cells care where nucleosomes are? Yes, they do. And look at gene yeast, they place an array of positioned at critical distance, not only from each other, but most importantly, transcriptional start. So here they are. These are just nucleosomes summed up across 5,000 genes. In particular, this transcriptional sits just on the edge of +1 nucleosome. And then you're upstream of a, and I think the pen's going to run out if we have a second pointer. I don't know. Okay. Well, I'll try and describe it with so stream of the nucleosome region right there. There we go. We have a nucleosome, which is where these regulatory elements are. Thanks. And downstream of the traditional site, down toward the 3' region of the transcription, and another NFR as well, nucleosome free. So you can imagine that the chromatin fiber, during the life of the cell and through particularly developmental stages, goes through a number of transformations, including the transcription. The gene have to be transcribed, DNA replication, recombination, repair. Oh, thanks. No, I got a second one. Thanks. Now we've got a bunch of them up here. All right, good. But in particular, one of the things I want to focus on in this talk is what happens as cells go through this enormous degree of compaction and stretching that takes place during one particular developmental stage in budding yeast, the sporulation process. Okay, now, before I get into that, since this is largely, I believe, a computational group, I just want to touch on the methodology so you see where some of this is coming from. And actually, one of the bottom lines is much of the methodology may be actually different from lab to lab, but essentially they produce very similar structures or maps of nucleosomes across the genome, which is essentially gratifying. We work with the budding yeast. Here's a cartoon of that fiber. We basically cross-link it in vivo, adding formaldehyde, and we believe it cross-links many places, but it doesn't cross-link every single nucleosome. It just, it's a stochastic process of what gets cross-linked and what doesn't. We then break open the cells by mechanical shearing, isolate the chromatin fiber, and then digest it with micrococcal nuclease. And this is just a section of the chromosome just showing MNAs chewing up to form these mononucleosomes. And this is what we go after. We purify those, immunopurification using antibodies against histone H3. And in particular, we do it under conditions that don't allow you to purify anything unless you have a cross-linker present. So we know that the mononucleosomal DNAs that we are extracting out are actually cross-linked to histone H3. Okay, and so ultimately this is the kind of distribution of tags. We do deep sequencing on that. And this would be one nucleosome here, another one, and so on. And what's really striking about this is here you see these beautiful arrays of nucleosomes and nucleosome-free regions at the beginning and genes. Strikingly, we see very, very, very few tags in these areas. We really believe that they are nucleosome-free, or at least less than 5% signals that we're seeing in genic regions. So they are really consistent. And ultimately, it leads you in a composite to produce these kinds of displays. Okay. If we looked at the nucleosome signatures, and John had mentioned about these dinucleotide patterns of AAs, TTs around the beginning of genes, we see, I don't know if you can see the color scheme there, but the reds, the greens, and the reds, each line here is a gene. There's about 6,000 of these genes. And we see strong correlations in red to nucleosome positioning sequences and anti-correlations in green. And so this is what relates to what John talked about in terms of encoding preferences for nucleosome organization around the 5' ends of genes. Okay, so now there's some practical considerations in this matter. We were concerned when we were first doing this, and a number of groups had come out with maps of nucleosomes. How robust are these patterns that we're seeing across the genome from different types of platforms, the hybridization type platforms, sequencing platforms, versus one lab to another, and also in terms of bioinformatic processing of the data to make nucleosomal calls. How similar were all of these? So there'd been a lot of research on this, and at the time we produced the study shown here and published in 2009, there were 6 data sets available across 5 different platforms in 5 different laboratories. So this was a really nice opportunity to compare. These are the 2 454, one of them from the Windham lab, one of them from our lab. Solid sequencing, Illumina, Affymetrix hybridization platform, and another Affymetrix custom array. All these cells were growing under basically the same conditions. So you can make nice comparisons. And we saw about the same number of nucleosomes in each case. Shown here is an alignment of those 6 different data sets around the transcriptional site. And you'll see is a remarkable, basically reproduce and recapitulate the same. These mapping are very robust, and thing that may appear different is that some of these data sets form stronger nucleosomes. Issue due to modeling that data. We believe that down towards the 3' well positions they see here, and so you're less confidence. And we've created a set and it's published that was relatively important. Think positions are going to play specific functions in specific transcription factors, for example, chromatin remodeler group versus another group are talking about the same position. And so that of fine position and we also produce a sort of a genome-wide browser map of this, which is available to this site. Here. Basically, what it is, these are tracks of individual data sets. These are you can load up and okay. So map reproducibility is really robust platforms, but there are some considering that there's a reference map that may be useful in comparing different experiments. Now we go on, and John showed you this picture. Thinking about chromatin inside a cell, curious as to whether action that it do followed by onto sporulation what might happen stay the same, or do they change? So make maps that work deal with cells haploid life cycle. In this stage, more in the cell cycle, the haploid version that goes to a cell, and also under a sporulation stage when in a carbon source. And so this is the stage, few minutes here, what's happening to the chromatin, particular. Okay, so what's going on during the stage? Now, I'll be focused on several stages. One is comparing the nucleosome organization in rich media. This is YPD media. And then we starve cells or put them actually on a non-fermentable carbon source for a while and then starve them. And so this is a carbon-nitrogen starvation point. This is the 0 time point. And then they start to go through a sporulation program, 2 hours, 5 hours, 7 hours, and 24 hours. Various steps of this process unfold. And this is where we're going to be generating nucleosome maps throughout these time points. So a couple of considerations. One is we have certain expectations of these cells, and we have to make sure that they conform to that. So we expect that during DNA replication that the histone levels double, and they do. They go up, and then they level off. Importantly, at the 24-hour period, our yield, this is what at 24 hours is when these cells have really formed spores. And our yield of histones is the same as at 7 hours, meaning we're able to actually extract out these histones and measure them, and therefore measure the nucleosome status of those cells. We confirm this through FACS analysis of them moving through the sporulation phases, and through visual inspection under a microscope going through meiosis 1, meiosis 2, and then ultimately under ASCII formation. So we detect over 90% of these cells moving through that process. So we think we have a fairly homogeneous population that we're examining here. So here's what it looks like in the diploid cell, just in rich media. You basically see that pattern that I just showed you before. That's to be expected because the patterns I showed you before were in rich media as well. Now we go under starvation conditions, and in light gray is the YP pattern, and at 0 hours is the first hour which it's going to proceed sporulation process. Essentially the same pattern retained there. If anything, you might see down towards about 1 kb downstream, the space actually increases in these cells as they undergo, when they're under this starvation state. But position plus 2 and plus 3 basically stay the same, the -1 deep NFR. I don't have to fill in of that. So it basically maintains its same structure. Now move out to 2 hours. You can barely tell the difference. These nucleosomes stay exactly the same. It shifts back in their position in terms of the spacing. We now jump out to 5, exactly the same position. The NFR is maintained, identical, nothing changing. And then all the way out here, where you might expect, okay, these things are now packaged inside, you might expect the far to fit in. You might expect positioning to be closer together. Maybe you wouldn't, it was really unknown. And I'm surprised to find it really nothing to keep the NR, and you keep those positioning. All right, so we're going to package all this in fiber, but we're going to keep primary structure. You might imagine it makes sense. These cells have access through replication, and unfurling the transcription programs at these stages. We then therefore rationalize a reason that the cell deals with all these issues. We're still curious to find out whether there were places where there occurring up of nucleosome-free regions. So with 3' end genes, we found no difference. All genes. So what you're looking at here is a row of 6,000 genes here, and this is the transcriptional starts. These are. You can zoom into any small. Nothing changes. Sporulation gene position in terms of occupancy level drops, where you might expect the gene to be turned on. So we do see the kind of typical things where nucleosomes get depleted when genes get turned on because the RNA describe that gene. But then those nucleosomes come right back to the same place. We looked at the ribosomal protein genes. Now, the reason why we look at this collection of genes is the huge NFR. The two nucleosomes here. Yet there is nothing detected when you go into the sporulation stages down by no nucleosome appears. So everything is largely fixed. You do see change in nucleosome occupancy level, as you might expect in low occupancy level because they're hypertranscribed, and then when they go stages where not transcribed, higher occupancy levels appear. We don't detect anything there. Changes there as well. We persist something called fuzziness, which actually Alejandro first described in the first paper on this, and that is the degree at which these nucleosome signals are positioned at focal point, center. So this would be a highly well-positioned nucleosome, and this would be a more fuzzy, less well-positioned. And we see something change over the course of the sporulation process. We thought it would change because, after all, this chromatin fiber is becoming more and more, and so you might expect positions of nucleosomes to be more restricted. They're not. We look at width of the nucleosome. Now that's the distance between a border measured via MNase digestion on one end versus the border measured on the other end. And typically it's about 150 base pairs in size. You might find some that are smaller than that due to chewing in, or some that are larger because a protein might bind there, like histone H1. Now, histone H1 is expected to help compact this genome as it moves towards the sporulation process. But we see no change in the width of those as well. So I think the point we're getting at here is that it's remarkable the degree of constancy of this chromatin fiber as it goes through some major transformations during developmental stages. Okay, now I wanted to pursue a different question because this was a question that was posed by an anonymous reviewer of a manuscript that we were trying to get published. And the reviewer said, well, why don't you look for changes in positioning around the transcriptional start site? So we pursued that, and because we hadn't thought of that, because at the time these structures were basically the same. So the question is, we have transcriptional start site here right on the edge of this +1 nucleosome. Okay? That's where it's supposed to be. But you can imagine small changes, and John discussed this and I've discussed it, that small changes in the positioning of this nucleosome could affect the utilization of a transcriptional start site here becoming more exposed, and in another situation, if it moved upstream, becoming perhaps more inaccessible. Now, I should point out that in multicellular eukaryotes, this is typically the situation. You often have a transcriptional start site that's unfettered by that first nucleosome, and this is what we see in yeast. So we searched for this, and the question is, do nucleosome positions shift? And if so, are there changes that are linked to transcription? So do you go from this status to this status, or this status to this, or any of these interchangeable forms during starvation or any of the transcriptional processes? So as you go from YPD to carbon, nitrogen starvation, are we seeing any of these? So we're going to report on this by color coding whatever we see. If a nucleosome shifts in this direction towards the downstream, we're going to color it yellow. So yellow meaning a more positive direction, shift downstream. And if it shifts in the upstream direction, we're going to color code it blue. And we're going to look at various stages of the sporulation process. Now, I'll blow this up in just a second, but this is what we're seeing here. These are about 6,000 genes. We're looking at the +1 nucleosome, and you see yellow here means it's shifting in the downstream direction, actually upon carbon-nitrogen starvation, and it doesn't change during the sporulation process. These genes here are shifting in the upstream direction. We don't see any of these cases here. In all cases, well, the vast majority of cases, what we see is it goes from either the canonical transcriptional start site to a position where the transcriptional start site is now further into the nucleosome, or to where normally this in YPD, and it then shifts down to the more favored position upon carbon-nitrogen starvation, depending on which gene. So we don't see any of that. Okay, so here's basically what I just described to you. Again, these yellow guys are moving downstream, and these blue guys are moving upstream. And the question is there any sort of linkage to some sort of function based upon those patterns? Turns out there is. We searched a number of sets to ask what might these genes be. They are linked to genes that are either induced or repressed starvation. So these genes here overlap carbon starvation genes. Okay, and if you take a look at those around, you can see that during the starvation and downstream, during sporulation, you have a shift down at that nucleosome position. These genes they actually move to the more upstream. So just to summarize, I've just shown what we're seeing here are genes that are enriched here. Okay, being shifted to state, the transcriptional start is now poised at the edge of that nucleus when these genes are going to be activated upon carbon starvation. There's another set of genes that are active, but get shut down as you move into the carbon starvation. So there appears to be some evidence here for regulation of transcription start site accessibility by repositioning plus nucleosome. And so there's, so yeah, the cells are where these are positioned, and they take advantage of that. Okay, so I'm going to wrap up here just to there's common organization exists around nearly all. There's a one position and NFR plus nucleosome. An array of nucleosomes downstream that there's evidence for just packaged in by principles of statistical positioning, which ultimately end 3' genes another nucleosome-free region. And so if you have these pattern nucleosomes that are highly regular, the cell is probably going to take advantage of both positioning of these nucleosomes and the factors that interact with specific nucleosomes. And so we may need a systematic nomenclature for defining which nucleosomes we're going to have in the genome when we look at factor nucleosome interactions. Surprisingly, the transformation that a chromatin fiber undergoes, the compaction, the expansion, all those kinds of transformations has very little impact on the structure. It attempts to maintain basically the same status. It really cares about where things are positioned because all these actions are presumably regulated at the point of accessibility to the DNA. So it needs to maintain that pattern. Positioning over the transcriptional start site may be of importance. Okay, the people who did this work, talented graduate student and postdoc, did the meiosis, Li Zhang, and Shizong Jiang did the systematic nomenclature mapping. All right, I think I'll stop there and address any questions that you might have.
Thanks, yeah, please.
Thanks, that was really interesting. I have two questions, one short and one a little bit longer. The first one is just, it's interesting to hear about this mechanism of shifting, and I'm wondering if there's a canonical distance, and if it's, I mean, I would expect it to be that the shift is usually multiples of 10 based on what we heard about the sequence pattern. Is that what you're seeing in the data?
We haven't looked at that yet. I'm not sure we have enough resolving power to determine that. So when we first produced nucleosome maps a number of years ago on a genome-wide scale, we also saw these 10 base periodic shifts. But that's most evident when you look at, you have the power of large number of nucleosomes looking here. Here we're talking about 80 to 100 genes, and you might not be able to get that kind of resolving power at this point.
And the second question related to something that you said in the very beginning, I didn't realize the cross-linking was so inefficient. It makes me wonder if there's a possibility for ascertainment bias there. Do you have any sense for whether...
What was that last word?
Any bias coming from the cross-linking? In other words, if there's three-dimensional constraints for how the cross-linking gets chosen, then quantitative assessment of how much nucleosome positioning would be difficult because of that bias.
Yeah, I'm not sure if there's any bias in particular as opposed to just random, you get a cross-linking or it cross-links with something else. We've looked at data sets where there's no cross-linking involved, and they look very similar to this. So we don't see, I think you can create these patterns without having a cross-linker present. But if you're looking at specific locations, then, so we're talking about bulk behavior, it doesn't appear to be different. But if you're looking at specific genes, there may be differences.
So Frank, do you know what's the end-to-end change in compaction during meiosis? So the question I'm asking is basically, does a chromosome go from a micron long to 0.5 microns? Or
It's about twofold, 2-3-fold compaction in yeast.
So,
in higher eukaryotes, it's much more compaction. But I think those are measured more of at an ultrastructural level.
So do you think what's going on is that now so one of the models for how chromosome compaction works in mitosis is sort of is it called axoneme, with a bunch of loops coming off of it? So you can have something that's not coming off loops on an axis that's fairly wide. And now if you just bunch it up, you can get pretty strong end-to-end compaction. Do you think that's what's going on in terms of compaction?
It could be. None of the experiments that we've done really address or can address the sort of the higher order structure on these. So your guess is as good as mine. It's what's happening at the higher order level.
Go ahead.
Well, there's a clear difference in the positioning of TSS and nucleosomes between mammals and yeast. So in mammals, the transcription starts is in the nucleosome-free region.
Right.
Do you have an idea or a hypothesis why this is the case?
Yeah, I sure do. That's a whole other talk. So we had mapped nucleosomes in Drosophila, and you see the same thing. And what you find is that the nucleosomes are downstream. The first nucleosome is downstream of the transcriptional machine. Initially, seeing where's the first nucleosome. What's going on in what